All right, ladies and gentlemen. So for this presentation, we're going to talk a little bit about the special variation of cell division that is meant specifically for sexual reproduction, known as meiosis. So to get into some of the details of meiosis, we need to discuss a couple of key ideas that go along with cells. So meiosis only applies to organisms that sexually reproduce. And that's going to be a key idea, all right? It only applies to organisms that sexually reproduce. So whenever we talk about meiosis, we are only talking about sexually reproducing organisms. And if we're talking about organisms that sexually reproduce, what we're actually talking about are organisms who must cut their chromosome number in half and then recombine it. So for instance, a human being has 46 chromosomes, okay? So two human beings want to reproduce. Well, each human being has 46 chromosomes. So what has to happen is this human being has to donate 23. This human being has to donate 23. And then they combine to become a new 46. That's half and half. Now, this has to happen in an orderly way. You can't just get whatever half of the chromosomes you want. It has to be the exact right half so that things match up properly. And so to do that, sexually reproducing organisms are what we call diploid. Diploid is the total number of chromosomes an organism possesses, a sexually reproducing organism. And what you will find is that for sexually reproducing organisms, all of their somatic cells, and somatic means body cells, so basically anything that's not a sex cell, are diploid, meaning that they have 2 times the actual number of chromosomes. And you can see that here. This is 1 chromosome. The diploid organism has 2 of them. This is 1 chromosome. The diploid organism has 2 of them. This is 1 chromosome. The diploid organism has 2 of them. And the reason that they have 2 of them is because 1 came from the mother and 1 came from the father. Now, when the organism is preparing to sexually reproduce, it has to take that diploid number and cut it in half. And so you'll see that here there's 1 red, 1 blue, and 1 yellow. And what's happening here is the organism is taking its chromosomes and putting 1 into a sex cell, so that way when 2 haploid combine, or 2N, together you end up with 2N, which is how you end up with 1 full human organism. Cells that are haploid are known as gametes, and they include both egg, or ovum, and sperm. All right, those are the only cells in the body that are gametes. So if you're ever presented with, well, what are somatic cells? The only thing that somatic cells are not are sperm and egg. Somatic cells are everything else but sperm and egg. All right? So let's talk a little bit about what's going on. So you see this diagram up here, and you have a cell, and the cell has 1 set of homologous chromosomes, the red and the blue. Now, you'll notice that I have the red sort of boxed off to let you know that the red is 2 sister chromatids, which we talked about in our last presentation. 2 sister chromatids are identical. They're just copies that were made during the S phase of the cell cycle. But the red and the blue together are homologous. They are similar. So that means that they have the same traits on them, but they can have different versions of the trait. Now, what happens for sexual reproduction is those homologs separate. So you can see that right here, I've got 2 copies of the red version of the chromosome, and over here I have 2 copies of the blue version of the chromosome. And then there's a second separation where I have 2 identical cells right here that are red, 2 identical cells right here that are blue, but all 4 are similar because they have the same traits. They just don't necessarily have the same versions. And so when we're talking about meiosis, or cell division for sexual reproduction, what we're actually getting at is that there has to be a special way to make this happen. How do you separate it so that you go from what is, at the start, a diploid cell, and end with what's a haploid cell? And so we go to our process of meiosis, all right? Now, meiosis has 2 stages. You can see right here we have meiosis 1, and right here we have meiosis 2. Now, these two stages have very similar processes to mitosis. And the reason for that is because meiosis is a evolutionary variation that's been made to mitosis. So first you have interphase. Cells that are going to enter meiosis start in some form of interphase, and then they enter the first stage of meiosis, which is prophase 1. Now, prophase 1 is very similar. Chromosomes shore up, so chromatin becomes chromosomes. And what happens in prophase 1, however, is crossing over happens. So we talked about that in our last presentation. Crossing over happens in prophase 1, and homologs pair up. So during prophase 1, the homologous pairs find each other. Now then, during metaphase 1, the homologs line up at equator, or at the center of the cell. So during mitosis, they line up single file. During meiosis 1, they line up in pairs. And that's the big, most important difference. During meiosis 1, they line up in pairs. Then during anaphase 1, the homologous pairs separate. So what that means is during telophase 1, the cell's already haploid. Each cell in telophase 1 only has half of the required genetic material. It has 2 copies of that half, but it only has half. Now, what happens after cytokinesis is the process repeats, and this is where evolution is weird. It takes the same process and just sort of modifies it. And so what happens is you enter the whole PMAT process a second time. Now, prophase 2, metaphase 2, anaphase 2, and telophase 2 all work just like mitosis. So during prophase 2, the chromosomes coil up into chromosomes themselves as opposed to chromatin. During metaphase 2, they line up, and you will notice right here, look, they're single file. Okay. During anaphase 2, sister chromatids are ripped in half. And then during telophase 2, the cleavage furrow happens so that the cell can split in two. The nucleus starts to reform, and the chromosomes uncoil. And then when you're done, at the end of cytokinesis, you have 4 haploid cells. Okay? Now, this is going to be really important. These 4 haploid cells. If there's sperm, there's 4 of them. If they're eggs, only 1 cell has a full cell body. Other 3 are just dummy nucleus. The egg cell has to have everything the cell needs. So when a new organism is developing from sexual reproduction, it requires an endoplasmic reticulum. It requires a Golgi apparatus. It requires mitochondria. If it's a plant, it requires chloroplast, vacuoles, lysosomes. And so all of that stuff has to come from somewhere. And for sexually reproducing organisms, that comes from the female. And so to make that work, instead of splitting all these cells evenly, they split 1 cell to have all of that material, but they still undergo the process of meiosis so that the nuclei divides properly. All right. So just so you're aware, sperm cell, 4 of them become functioning haploid gametes. For eggs, it still does the same exact process, but at the end, in something that we call oogenesis, 1 functioning egg cell is made, which we call an oocyte, and then 3 sort of dummy cells are made. All right, that's it for mitosis or for meiosis.